I want to start this award presentation by thanking ASLO for organizing this online ceremony and also for giving me the chance to provide a presentation through this route. It's a little bit awkward from my home office, and I really hope we can meet soon at an upcoming ASLO meeting. I also really want to thank the award selection committee and of course the nominators for granting me this award. It really means a lot to me, so thanks very much. And yeah, this award and my work was not possible without the many collaborations I have with like my mentors, colleagues, and a whole number of students in my lab. And I want to start by thanking my PhD advisors, mentors during my PhD from the University of Amsterdam, Jeff Huisman and Petra Visser, and from the Netherlands Institute of Ecology, Ellen van Donk. They really showed me how to turn ideas into testable hypotheses and then bring this into experiments and really test this experimentally. And yeah, I really learned also from Jeff and Petra how to connect this work to theory, particularly related to cyanobacterial blooms. And also Ellen showed me that, well, ecological interactions from grazing and parasites really matter as well. So thank you very much. Then I also want to thank my advisors during my postdoc, which I did at the Alfred Wegener Institute, so Björn Rost and Uwe Joon, who really showed me more about the marvels of the cellular processes, so carbon acquisition and a whole lot of the molecular ecology of harmful algal blooms. So thanks guys for that. And then there are many people in my career that really helped me by inviting me for special issues or inviting me for a talk. Or even for workshops, which really meant a lot to me. And as an early career scientist, these gestures really mean a lot. So thanks all very much for this. And then of course, all the people in my group that have been in my group in the past, visited my group, are in my group now. So all the PhD students and postdocs, thank you all very much for your motivation, your energy, and yeah, this really inspired me a lot through times. And today I'm really grateful to my department, so the Aquatic Ecology department at the Netherlands Institute of Ecology. Yeah, this is a wonderful group of people, my colleagues, senior scientists, the technicians, and a great, enthusiastic, motivating team of PhD students and postdocs. So thank you guys very much. For this talk, ASLO asked me a few questions for the ASLO bulletin. So, and I thought these questions are nice to actually use for this talk as well. So the first question was, what would you say has been your most exciting finding so far, and where do you envision your research program going in the future? The next one deals with addressing the harmful algal bloom issues with the community, and what has been my greatest challenge in outreach. And the third point I want to highlight today is what advice I could give for students and postdocs on balancing the responsibilities for that you come across as early career scientists. So I will touch upon these during my talk here. So in my work, I'm interested in understanding harmful algal bloom dynamics. So here we see a cyanobacterial bloom in a Dutch lake, and I'm really interested in understanding what happens at this ecosystem level by looking at the community, at the population level, and also at the cellular level. And to be able to link cellular processes to the dynamics that we see at the population, community, and ultimately the ecosystem level. Well, to do so, well, we need theory, right? Because how do we cross these skills? That's a huge challenge in ecology, of course. And among the theories that frameworks that I have been using is ecological stoichiometry. Thanks Bob and Jim for writing this book. It came out three years before I started my PhD, so that was a good timing for me to start, and I was heavily inspired. And well, I also put a picture of my own book here, and you see it has been read. I must admit it's one of the few books I really read from head to toe, so thank you very much. Then I also came across literature, and when starting my PhD, I was interested in cyanobacterial blooms and toxicity of blooms, and I came across literature in the terrestrial world, and there has been the carbon-nutrient balance hypothesis cited a lot and used a lot, and it has been declared dead and resurrected again. So it's a highly debated theory, but the basic idea I thought was kind of interesting where it describes how metabolites, secondary metabolites that can be carbon-based or nitrogen-based, are regulated by light or nutrients. And this, of course, matches very closely to the ideas presented in ecological stoichiometric theory. And if we look at the toxins that are produced by cyanobacteria, these are heptapeptides, so they consist of amino acids, and they vary in the types of amino acids on 2 positions. So depending on the amino acids incorporated, this compound can be more or less nitrogen rich. And we were wondering whether this compound as a whole, because it's a nitrogen rich compound, but also the composition can change in response to the balance of nitrogen and carbon. And this is also what we've shown. So we performed chemostat experiments, and here we see the microcystin content, in this case microcystin RR, which is the compound, the analog with most nitrogen in it. And we see that it responds to the cellular N/C ratio, where low N/C ratios under nitrogen limitation also led to low levels of these microcystins. And if we had light limitation, then we had higher N/C ratios, and we see an increase in these microcystin levels. And carbon limitation did increase the N/C, so it still had a high N/C ratio, but apparently there was not enough carbon available to produce these compounds. So this partly matched the hypothesis that we would expect that these nitrogen-rich compounds follow the N/C ratios. And we also showed this in the natural environment, where actually the relative content of microcystins, so of microcystin RR, this nitrogen-richest analog, varied with the N/C ratio. So higher N/C ratios generally corresponded to a high contribution of this microcystin RR. And the ranges were quite comparable between the lab and the field. So that was quite kind of exciting. For me, at least, it was one of my first publications, and I was really inspired by the idea that we can look beyond our field and learn a lot from ecological concepts that are out there and try to bring those together. Then, of course, we repeated this. Well, many people performed these experiments, and what we did is actually take this data from literature. So what we looked at is looking at the microcystin contents in cyanobacteria, and well, mostly toxin content in dinoflagellates, which are the marine species. And we looked at these nitrogen-rich compounds and then how they were regulated in response to limitation of nitrogen and phosphorus, and thereby shifts in the stoichiometry. So that's what we see here on the y-axis is the toxin contents, the change in that. So if it's positive, there's an increase. If it's negative, there's a decrease. And on the x-axis, we see the response of nitrogen to phosphorus ratio. So if it's positive, there's an increase in the N/P ratio, and if it's negative, there's a decrease in this ratio. And what we see is quite consistent that under P limitation, we see generally an increase in N/P ratios and also an increase in these nitrogen-rich toxins. And then with N limitation, we see a decrease in these N/P ratios and also a decrease in toxin levels. So this is what we have shown a couple of years ago, and well, quite a lot of literature came out in the meantime. So we performed again this analysis and well, show very consistent patterns throughout. Then, of course, these are responses in elements, and underlying these elements, we have biochemicals, right? So we also, if we look at the biochemicals, then microcystins, as I said, is a heptapeptide. It consists of amino acids. So we would expect, and it's also what we've shown, is that microcystin content as a total really follows the amino acids. And since these are nitrogen rich, they really follow the nitrogen balance of the cells. Then in dinoflagellates, we show the same thing. So PSP toxins are produced by both dinoflagellates and cyanobacteria, but this is data from dinoflagellates in the marine system. And PSP toxins, it's mostly saxitoxin and different analogs. They use arginine, which is a nitrogen-richest amino acid, as a precursor. So it really relies on arginine, and that's also what we showed here. And arginine is very nitrogen-rich, so clearly PSP toxin content follows this arginine. And therefore, the relationships between the nitrogen levels and the toxins we can really explain by the underlying biochemicals. So I think this package is something that shows how we can apply ecological theory to these kind of very bioactive secondary metabolites that are really important for water quality and also with consequences for ecosystem services, right? Because we do use the lakes for many things. So this is a really direct connection between, I think, fundamental sciences and ecosystem services. Then what's next? So I'm already working part on that, and I'm very interested in looking further into the regulation. So we can use a lot of omics approaches, so metabolomics, transcriptomics, and see and try and further explain these responses that we see between stoichiometry and toxin levels to better understand what drives this toxicity of blooms. Also, we can look at different genotypes, so there are a lot of different genotypes, both in harmful algal blooms and cyanobacterial blooms, and we can look at these different genotypes, and they differ in toxicity as well. So either because of the amount of toxins that vary, or the types of toxins that are produced, so the different analogs. And by understanding the genotypic variation during a bloom, we can better understand bloom toxicity, and also we can better understand whether blooms can adapt rapidly, so throughout the season, if there's rapid evolution, for instance. So those are some thoughts I want to explore further as well. And also I want to look at other interactions. So for instance, with viruses such as anaphages, we did some work on that already, and also fungal parasites, because many cyanobacterial species are not very well edible. They are not very suitable food source for zooplankton, but then they may be very well controlled by parasites. And I'm quite interested in their, especially their stoichiometric interactions. And lastly, I want to scale up further to the ecosystem level. So really looking at well, our large red lakes. We're a small country, so these lakes are still relatively small maybe, but for us they are huge. And I'm interested in understanding the dynamics that we see in these large scale systems, and especially by also start looking at from space, so using remote sensing technology. So those are just a few things I'm interested in exploring further, and to really expand this range that we cover, these different scales that we cover in understanding blooms, and in this case, cyanobacterial blooms. So then I'm also very interested in bringing out the science that we do. So I'm very excited always when having students in our lab, and especially if we can do experiments. So if there's enough time in the program, so we have them for a couple of weeks, and they can really start asking questions, but also answering questions themselves by performing experiments with phytoplankton. And yeah, we also often, I see a change in how they proceed. First, they are there because they have to be there, but in time they really start asking more and more questions because they see what's going on, and that's really something I want to expand further. Then I think also bringing out things to the media, I think that's super exciting, just to be able to bring out the science that we do, and especially what I'm interested in is bringing this curiosity that we have as scientists, right? Why do we study the things we study, and what is so exciting about ecological stoichiometry, or understanding the toxins? And of course, that's also important to know that, because society directly is influenced by these toxins, for instance. And I'm also very interested in bringing out the risks, because for some aspects of especially the marine harmful algal blooms, it's not very well covered in the Netherlands in terms of the risks are not very well brought into the picture. So we know very little about that. So by using the media, we can really bring out this risk and try and study that further. Then lastly, I'm also chair of the Dutch Cyanobacterial Platform, which is a group of experts working on cyanobacteria, both from science, but also from management. And this has been a very motivating group, because people bring in different expertises, right? To really see what's going on, what's the real problems, and how can we try and solve that, especially also using scientific approaches. So collecting data together, looking at the data together, and then inform policy about that. And well, this year we also advised on a new protocol in which the Dutch water boards will then use for monitoring and risk assessment of cyanobacterial blooms. And that's of course very rewarding as well. Well, lastly, some advice for the early career scientists. I feel I'm also very early career. I'm also learning this still now a lot. So I'm still looking around a lot and see how to deal with these issues that we come across during our career. And well, some things that helped me, especially over the last years when a lot of things came together, is well, the first point is prioritize and focus. I think, well, there's always exciting stuff coming, right? If you're interested, if you're curious, there's always new questions arising. But still, too many nice things still means too many. And I think I'm trying now also myself to be more selective and say, okay, this is something that really makes me tick faster, and that's really what I want to follow up further. So that's really prioritize and focus and act to that. Then I'm not sure if, I guess this is a bit kind of standard thing, but it really worked for me. So I block and use my golden hours. So I'm, for instance, very active in the early morning. So I really block my mornings and try to make my meetings as much as possible in the afternoons. This often does not work, but if I have 1 or 2 mornings in a week, that at least gives me time to write my manuscripts and proposals. And importantly, it's really important to communicate these hours to others so that people know that they don't necessarily, if it's not necessary, disturb you. And of course, writing there is super important. To continue writing manuscripts, it's not to publish, but to really bring out the work, to bring out the ideas to so everyone can read it. And it goes through a peer review process. And I think that is, it can be frustrating, but most of the times, actually, I feel that it's super rewarding because, well, clearly there's a publication, but it's also to see many reviewers are actually reading the stuff very well. Your colleagues really take time, and you can really improve it on the way. So I think that's very important about that. Then give responsibility. That's what I'm learning now quite a bit. So taking responsibilities is of course one thing, but giving them is another. It really helps to well be clear that others can also take stuff out of your hands. And related to that is ask for advice. So when challenged by these many responsibilities, you can ask your mentors, your colleagues, and students for advice, because many people have dealt or are dealing with similar issues. And yeah, by asking help, there's always help out there, right? And people are really willing to share their experiences. And I think that is something which is maybe a bit more difficult now in the days of COVID, of course, with many people working at home. And what helped me a lot is actually, well, to get inspired is important, but also when I looked at the ASLO website and at the meetings, it's just very hopeful to see that there are already meetings planned for the next 4 years. I mean, I'm sure we will meet at some point at the ASLO meeting, and ASLO is really taking a lot of effort to keep this going. So I thank ASLO again for taking all this effort. And yeah, I really hope we can meet as soon as possible. Thank you very much.